Alright, this is Intro Bio lecture number 6, and today we'll be talking about photosynthesis, which is another process that I've touched on a bit but never went into great detail about. It's usually associated with plants because the pigments used in photosynthesis are why many plants are the color green, but they also can be other colors. However, the first organism to conduct photosynthesis was actually a prokaryote called the cyanobacteria. And on that note, let's move on to the details of the actual process. Okay, so the overall goal, I guess you could say, for photosynthesis is to convert light energy to glucose, which is the sugar broken down during cell respiration. The equation for the process almost seems like the opposite of cell respiration, as it requires 6 molecules of carbon dioxide, 6 of water, and then some energy in the form of light, and this all outputs glucose as well as 6 molecules of oxygen gas. And the parallels stretch even further. Photosynthesis occurs in an organelle called the chloroplast, which is strikingly similar to the mitochondria. If you remember from a few lectures ago, I talked about the theory of endosymbiosis and how it states that mitochondria were likely the result of a prokaryote taking residence in a eukaryotic cell, and then them both kind of helping each other out, which is called symbiosis. So this resulted in the prokaryote staying and just becoming another organelle in that eukaryotic cell. Well, the chloroplast was also likely a prokaryote like that, but in this case, specifically a cyanobacteria that had already developed the process of photosynthesis. Therefore, the cell that adopted the cyanobacteria would be the ancestor of all modern plant life. Anyway, knowing that, let's take a look at chloroplast structure. First of all, it has a double membrane, so not one but 2 phospholipid bilayers, and just like in mitochondria, the intermembrane space is what's in between these 2 membranes. Now, the main structure in the chloroplast are the thylakoid membranes, which are these lighter green stacked structures, and they come in these stacks called grana. Finally, the gel-like substance between the thylakoid membranes, which is really everything else in the chloroplast, is called the stroma. Now, photosynthesis can be divided into 2 broad sets of reactions that both really consist of a ton of smaller reactions, and they're named after whether they rely on light or not. So they are the light-dependent reactions, which come first and need light to work, and then the light-independent reactions, which don't need light but rely on the products of the light-dependent reactions. Alright, now it's time for a really quick problem to test your understanding of this lesson so far. Photosynthesis is a blank reaction, endergonic or exergonic. I'll give you a few seconds, you can pause the video and work through it, then I'll go over the answers. This should be pretty straightforward. In photosynthesis, energy is added and bonds are formed to create the pretty large product of glucose. So it's definitely an endergonic reaction because once more you are putting energy in and getting chemical energy, or that same energy as chemical energy, in the form of the chemical bonds in that glucose molecule. So definitely endergonic. Alright, so before actually getting into the light-dependent reactions, I want you to have a good understanding of light as a whole. You can think of light as a wave, and the one important number to remember for waves, at least in this case, is wavelength, which I've marked off in these waves. Basically, it's how far the peaks are from one another. So essentially, the smaller the wavelength is, the higher the energy is. That's another important point to remember. So the top wave here has a really, really big wavelength, as you can see. That line which connects the 2 peaks is really long. And we know this because the distance between the peaks is huge. So this also means that it has low energy because remember, high wavelength means low energy, low wavelength means high energy, which of course also means that the wave at the bottom with a much shorter wavelength has really, really high energy. Now, plants want to absorb the energy in light and then use that energy to eventually make glucose. The way they do this is with molecules called pigments like this one. This is chlorophyll A, and to put it simply, it gets energy from light when light waves hit it and are absorbed by some of the bonds. Now you may wonder, what range of light can chlorophyll absorb? Well, the way it works is that any substance, including pigments, absorbs all the light that it doesn't reflect. And chlorophyll is actually one of the pigments that makes plants green. So this chlorophyll is absorbing more or less all wavelengths of light other than the range that corresponds to green, because it has to reflect that light for the plant to look green. This means that pretty much every color in the rainbow, except for green, is being absorbed along with some UV or ultraviolet light, which is light that has a higher wavelength than violet. And it's also why humans get sunburns, because you get that light from the sun. Interestingly, plants don't only produce green pigment. There is an entire class of pigments that range in color from red to orange called carotenoids, because the reason carrots are orange. Okay, now let's get started with the actual light-dependent reaction, which basically converts light energy into chemical energy, or energy stored in bonds. And this chemical energy is stored in two ways. One is ATP, a molecule we're very familiar with at this point, and the other is NADPH, which is very, very similar to NADH, the electron carrier used in cell respiration, except it has an extra phosphate group attached to it. So the full name is nicotinamide adenine dinucleotide phosphate, as opposed to nicotinamide adenine dinucleotide, which is what's used in cell respiration. In terms of location, the light-dependent reactions occur in the actual membrane part of the thylakoid membranes, and they occur through a series of protein complexes that are embedded in that membrane. The first two big ones are called photosystems 1 and 2, also known as P700 and P680, but those names aren't at all important to remember, and I'll go over what the photosystems are, their structure, and how they're different from one another in a minute. There's also an electron transport chain, which serves the pretty much same role as the electron transport chain in cellular respiration, except in this case, it's a different set of proteins and it's split up a bit, as you'll see in a minute. The transported electrons are used to convert NADP+ to NADPH, which, remember, is just an electron carrier along with to pump protons inside the thylakoid membrane to make a similar chemiosmosis system to the one used in cellular respiration. Finally, ATP synthase is used to make ATP using the protons pumped by the electron transport chain. So those are the four parts of the light-dependent reaction. Next, we're going to take a look at the photosystem structure. So as you can see, photosystems are pretty intricate complexes of proteins. The one you're looking at right now is actually photosystem 1, and I'll explain how you can tell the difference between them in a minute, but first let's take a look at what's happening here. These light green dots are actually chlorophyll molecules, and they have the ability to absorb the energy from light, as I just went over in the slide covering light. This energy is then transferred to electrons, which can then basically hop from one chlorophyll molecule to another until they end up on these two central chlorophyll molecules, which are surrounded by blue here, and these are called the reaction center. So basically, you can think of this photosystem as kind of like an energy funnel. All of these individual chloroplast molecules absorb light and then funnel all of that energy gained from absorbing the light into these two central molecules of chlorophyll using electrons. And from there, these electrons are passed onto this orange protein just above them, and then from there passed onto the electron transport chain. Anyway, this is photosystem 1, and the only difference between it and photosystem 2 is that while both can get electrons from absorbing light energy, photosystem 2 also gets some from breaking down water into hydrogen and oxygen, which you'll see on the next slide. Okay, this is an overall setup for the light-dependent reactions. As you can see, there's the two photosystems, but counterintuitively, photosystem 1 actually comes after photosystem 2. So, like I said, photosystem 2 can get electrons from both light, as always, and also from the breaking down of water, as you can see from the reaction down there. And those electrons are then transferred to the first part of the electron transport chain, which uses their energy to pump some protons into the thylakoid before passing all these electrons to photosystem 1, which then takes those electrons and also gets some more by absorbing light, then passes all of them onto the last part of the electron transport chain, where they are used to convert NADP+ to NADPH. Finally, the protons that were pumped earlier are used by ATP synthase to make ATP, and I think that about wraps it up for the light-dependent reactions. So the overall products are this NADPH and ATP. Next, we have the light-independent reactions. Remember that while these technically don't require light, they do need the NADPH and ATP made from the light-dependent reactions. Now, the light-independent reactions occur in a cycle, just like the citric acid cycle in cell respiration, so they are called the Calvin cycle. As you can see, the cycle is split into three general stages, namely carbon fixation, reduction, and regeneration. Carbon fixation is when carbon dioxide comes in, it's attached to a molecule called rubisco and converted to something called 3-PGA, a 3-carbon molecule that has a phosphate group attached to it. Next, another molecule of ATP is used to add another phosphate, and then NADPH and the removal of one of those phosphates is used to create a molecule of G3P, which can be used in a different pathway to make glucose. Remember, G3P was a part of the glycolysis pathway in cell respiration. But not all the G3P is used to make glucose. In fact, roughly 5/6 of it goes back into this cycle, where another molecule of ATP is used to convert it to rubisco that can then be used to make even more G3P. Alright, so this is an overall summary of the entire process of photosynthesis. The first thing to remember is that there are these two very separate and broad parts, the light-dependent and the light-independent reactions, and they both occur in the chloroplast. The light-independent reaction uses water and light to get electrons through the photosystems, and those electrons are then used to make ATP and NADPH. The light-independent reaction occurs as a cycle in which carbon dioxide is bound to rubisco, and then NADPH and ATP are used to make G3P, which can then be used to make glucose at a later point in time. Alright, this is my reference for the lecture, and I think that about wraps it up. See you all next time.